Hi, in today's lecture we are going to discuss about the interpretation of Franklin's X-ray diffraction pattern of DNA and how this interpretation paved way to Watson and Crick's model of DNA. But first things first, before I proceed further, I'll show you a basic diagram of how a helix looks when it is subjected to X-rays. You see here. On diffraction, a helix shows a pattern. uh, on diffraction, it shows a pattern like this. You see this X shaped pattern with all these stuff, which is due to the different planes of helix. You can see here, like when a helix is seen from the front view. From the front view, you can see there are presence of various planes. The helix is divided into various planes, which are placed at an angle with each other. This angle alpha, and also there is an angle between the pitch. This angle. Okay. So this diagram I am showing it to you because I won't be going into the physics of this, and these two diagrams overall will be forming a base for interpretation of the structure of DNA. Now coming back, we see a very symmetric pattern of X-ray diffraction of DNA. So one by one we will be studying about the nitty-gritties of this pattern. So here you see that when DNA fiber is subjected to X-rays, how it is being recorded on the film. Okay. Yes. So as you see an X-like structure in the film of the X-ray diffraction, how has it come out? Now you remember I showed you a diagram of how a helix looks like a look shows a cross-like pattern. Shows a cross-like pattern. Sorry. Just a minute. This, even if you don't look at this, yes, shows a cross-like pattern. Okay, X-shaped pattern. This X shape indicates that the dimensions of the DNA, that is the diameter and the radius, is consistent throughout its structure. See, it is consistent throughout its structure, and it also corresponds to the double helical structure of DNA. This X pattern guided Wilkins and Franklin to derive some basic dimensions of DNA structure. Now this X pattern is formed by the layer lines. This we call as layer lines, which corresponds to one turn in the helix, which is 34 angstroms. Okay. Uh, See here. This each each layer line is present at a distance of 34 angstroms. Now you may get confused that it might correspond to the base pairs, but it is actually not so. It is one of the basic principles of X-ray diffraction that closer the spots they look, larger the distance is present between them. I had shown you in the diagram which had the basic structure of a helical X-ray diffraction. Okay. So try to correlate with that. These layer lines correspond to one turn. This is, you see here, this is one turn of the helical structure of DNA, which we also call as a pitch, and its distance is 34 angstrom. Now coming back to this. Now you might again get confused about the distance from the center. This is the center to the dark spot over here. You may see here a very dark spot over here, which is actually 3.4 angstroms. This was the distance between each layer line was 34 angstroms, and this is 3.4 angstroms, which is quite paradoxical if we see it as a layman. But again, you try to refer to that structure which I showed you earlier, which had a little bit of mathematics involved in it, but don't go deep into it. This is actually the derived from the rules of X-ray diffraction, and as I told you earlier, I won't be going into the physics of it. So this actually indicates the distance between two stacked base pairs, or the vertical distance between the two base pairs. Now here you see one thing, you notice one thing, that in the that a pitch whose length is 34 angstrom and the distance between each stacked base pairs is 3.4 angstroms. So as we can calculate that in each turn, that is in each this one pitch, there are 10 base pairs. Okay, the pitch of the helix or the degree of rise can be calculated with the angle with which this X pattern makes with the horizontal axis, and there are some missing layer lines as we go up in the this X-ray photo. You can see here this is missing. At first it is a little bit consistent, but as you go upsides or this downwards, you will find it a little bit inconspicuous. Okay, it indicates that DNA double helix, though it is present, but it is present in an unsymmetric fashion, and it gives rise to two types of grooves, that is, this is a major groove and this minor grooves, and the spacing between the spots, this space between the spots will give us the helical radius and hence helical diameter. Because you see, this cross indicates what? Each turn. Okay, so each turn can be assumed as a circle, and these dark spots, they give a dimension of radius, and from radius is 10 angstrom. From radius we can calculate diameter, that is 20 angstrom. Okay, and from this diffraction pattern it was also deduced that phosphate moiety, which is present in the backbone, lies outside on the DNA of the DNA backbone. So that's all for today. I hope you like the video. All your suggestions, your comments, any sort of feedback is welcome in the comment section. Thank you so much for your patient listening.